Good evening everyone, welcome back to my channel. And in today's class, very quickly we will solve two questions that are very important for any type of competitive exams, biotechnology exams, any type of competitive exams like NET, GATE, and the upcoming JENPAS-UG 2025 also. And for that we are going to discuss this important topic. So firstly, see the first question that has come. JENPAS-UG 2023, that golden rice developed by genetic engineering is able to produce excess amount of what? There are the options like vitamin D, beta carotene and ferritin, biotin and vitamin C. So the correct option is vitamin beta carotene and ferritin. Okay, so answer B is the correct option. Why? Because golden rice, it is a genetically engineered plant and it's produce beta carotene, which is a precursor of vitamin A, and vitamin A is very important in the rice endosperm. So some advanced versions have also been modified to include ferritin, which helps increase iron content, and iron is the main component of hemoglobin, our red blood cell, right? The topic genetic engineering is very important. Okay, next question, that is the bt cotton. It is very important, bt cotton. There is a very simple question that comes JENPAS-UG 2024, that what is bt cotton? We all know that answer, that it is a genetically modified plant and can produce the natural insecticides. So it act as a pest control. Okay, why? Because bt is bacillus thuringiensis. It's a bacteria, but this bacteria produce a type of bt toxin, and which is very much toxic for some insect, some pest, okay, like bollworms. So see how, what is the principle it follows and its mechanism of action. So here is the process of creating bt cotton. Firstly, we need to know the genetic engineering, the process, how we can create bt cotton. bt cotton means, bt means bacillus thuringiensis. So bacillus thuringiensis produce bt toxin, and we are incorporating this bt gene with the cotton plant. It's a plant and it's a bacteria, so we are incorporating bacterial, a specific type of bacterial gene which is responsible for producing bt toxin into the cotton plant, so that this plant can prevent itself by getting harm from the insect. Okay, is it clear? Now there is a bacterium, that is the bacillus thuringiensis. It's a genome where we can see that the bacteria is having DNA, and it is also having a protein, that is bt toxin, and it is in crystal form. Okay, crystal protein, and as well as it's also having cell membrane, cell wall, normal property of a bacterial cell. For bt gene production, we need to extract this DNA from the bacterial cell. Okay, so we extracted that DNA, and this DNA is getting splitted by the restriction enzymes. Okay, and we identified the bacillus thuringiensis gene within that DNA. We have to identify that gene within that DNA. Okay, so this is the bt toxin gene, which has been identified by the help of restriction enzyme from the DNA of the bt, that means bacillus thuringiensis. Okay, DNA is housing a chain of gene but in that gene library, we need to choose which gene is responsible for making the Bt toxin. Okay, so that is the Bt toxin gene. Now we are going to transfer this Bt toxin gene into this plant, that is the cotton plant. So we cannot transfer that bacterial gene into the plant cell. For that transfer, we need a transfer element. We need a vehicle from one place to another place. For visiting one place to another place, we always travel by any type of vehicle. So here is the vehicle that is named as Agrobacterium tumefaciens. This is the vector. Okay, and this vector is having plasmid. Plasmid is removed from the cell, and we cut this plasmid by the help of restriction enzyme. Okay, and we incorporate the Bt gene into that plasmid. Bt gene into that plasmid. Okay, plasmid is combined with the Bt toxin gene. After combining Bt toxin gene with that plasmid, again we are placing it back to the within the Agrobacterium tumefaciens. Okay, and then these vectors transfer this plasmid, this DNA, to the plant cell, where within the plant cell we are now having the Bt toxin gene, and this plant cell are going to produce the pesticide. Okay, Bt toxin, it's producing Bt cotton. That is how we have created this Bt cotton plant. Next, the mode of action of Bt, how it actually works. So the mode of action of Bt involves a specific biological process that targets insect pests primarily in orders to Lepidoptera, that means caterpillars, Diptera, that means flies and mosquitoes, and also Coleoptera, that means beetles. Okay, but is a naturally occurring soil bacterium. It's a naturally occurring soil bacterium that produce crystals. Which type of crystals? Like the proteins. It's a crystal cryproteins, and cryproteins which are toxic to the certain insects. Okay, so basically what happens that Bacillus thuringiensis produce proteins that kill certain insects such as lepidopterans and the coleopterans, that means the beetles, and also the diopterans like the flies and mosquitoes. Okay, but Bacillus thuringiensis forms a protein crystals during a particular phase of their growth, and these crystals contain toxic insecticidal protein, that is the cryproteins. Okay, that is the cryproteins. Now the question is, why does these toxin not kill the bacillus? Because Bacillus thuringiensis, it's a bacteria, and it is also producing like this toxic substance crystal cryproteins. So why they are not getting killed by this toxic substance? Okay, if they are also producing this toxic element, because Bt toxin protein exists as inactive protoxins. It is exist as inactive protoxins forms. Okay, protoxins, but once an insect ingests, the inactive toxin is converted to its active form due to the alkaline pH of the gut, which solubilize the crystals. That is why there is the first step, that is the ingestion by target insects. Okay, when the target insects, maybe the beetles, caterpillars, diphtheria, any of these insects ingest any part of the bt cotton, then that cry proteins get activated, within their gut. Okay, where they get activated? Activation in alkaline pH within the caterpillar body. Okay, because that caterpillar ingested that bt. Okay, caterpillar ingested that bt. Now these bt, that crystals, that cry proteins are within the gut of the caterpillar. This is the gut of the caterpillar. So within this gut, the pH is alkaline pH, not acidic. It is alkaline. So these crystals get activated in this alkaline pH. Okay, and what they do after activation? They bind to the receptors. And where these receptors are located? On the epithelial cells of the midgut. So they binds, cry toxins binds to the specific receptors. What type of receptors? Cadherin like receptors, aminopeptidase and alkaline phosphatases. Okay, and what they do after binding with the receptors? They form pores. That means they create holes within the caterpillar body. Within the caterpillar body, they create holes. That means if this body is here, they create pores by the help of these cry proteins. Okay, if they form these pores, what happens? Which disrupt the osmotic balance. So cell lysis and gut paralysis. The disruption causes cell lysis, that means bursting, gut paralysis, leakage of gut contents into the insect's body cavity. And also there occurs septicemia and death. Okay, and important note that this bt, that means bacillus thuringiensis, this toxin is also that it do not harm humans. We are safe, animals are safe, and it is also safe. Bees are also safe from bt. Okay, bt genes like cry1ab have been genetically engineered into crops like bt cotton and bt corn. These two type of plants developed by bt genes. Okay, and here is the picture of the whole story, that this is the caterpillar and it has already ingested some part of the bt. Okay, that is why there is the protein, crystal protein, and second step is the dissolution of protein crystals. These protein crystals dissolve within that pH, that is the alkaline pH, and they get activated into the alkaline pH. And what they do? Activation of protein, that means the proteins are delta toxin and toxin. They bind to specific receptors. Okay, so if it is a caterpillar body, if it is a body of a caterpillar, okay, and there is the presence of these cry proteins, and they get activated by the pH, alkaline pH, and this is the gut of the caterpillar, any insect's gut. Okay, and there is the epithelial cells, and these epithelial cells houses receptors for cry proteins. There are the receptors. So the cry proteins bind with the receptors, and what happens? It creates pores here. So the membrane, cell membrane get disrupted. This things occurs, and that is why this whole insect get dead. Okay, so this is the scenario of bt cotton, and this is really very important topic. Do not skip. Stay tuned with me for such type of videos, important videos, and don't forget to state your comments, and it becomes helpful for you or not, let me know. And thank you.